Ganz, Ganz from the University of Copenhagen, and she's currently a researcher at the Neurobiology Research Unit at Rigshospitalet in Copenhagen, and she's an associate professor at the Department of Computer Science at the same university. As a researcher, she has an interdisciplinary background based on physics, computer science, and neuroimaging. Her research focuses on the application of medical imaging processing and machine learning to clinical medical image data, mostly neuroscientific data. Additionally, she aims to embrace open science principles in her research. Hence, she's involved in data sharing initiatives such as the brain imaging data structure. The talk will be continued by Cyril Pernet, who is also working at the Rigshospitalet and the University of Copenhagen. Cyril achieved his PhD in neuropsychology at the Université de Toulouse-Le Mireille, with training and employment in renowned institutions such as INSERM in France, the Low Temperature Laboratory in Helsinki, the Center for Cognitive Neuroimaging in Glasgow, and the Center for Clinical Brain Sciences in Edinburgh. Cyril is an experienced academic researcher and scientific project manager with a long track record about multidisciplinary research in brain health data science, including cognitive neuroscience and neuropsychology. He has also expertise in biomedical imaging technique, including MRI, functional MRI, EEG, PET, with a strong history of research and development of digital signal processing, image analysis, and statistical modeling. He has a leading expertise in brain data international standards for curation, sharing, and EU GDPR compliance. Now, the floor is all yours, and Melanie Ganz will start with her talk.
Thank you very much for the introduction. The aim of today is that Cyril and I want to introduce you a little bit to the open science efforts that are going on in the PET neuroimaging community. And when we're looking at this, I wanted to take the time and introduce you first of all to the brain imaging data structure. What is the brain imaging data structure at all? And it's called brain imaging data structure, but it's not a new data structure per se, so not a new imaging format like NIFTY, Analyze, DICOM. It's more a way how to structure and name your data. So it's more about how you organize data in folders, how you name your files, how you document your metadata, and above all, how to use community standards and dictionary to do all this, so that the community has agreed on it, so we don't use idiosyncratic naming, for example. It works in general, or was developed in general for imaging data, but by now it is also extended in a lot of different areas that I'll highlight to you in a second, and it also covers behavior and cognition. And you can access all the documentation about BITS and get more information on bits.neuroimaging.io. And a lot of those BITS slides, introductory slide, I actually got originally from Cyril, so they are stolen directly from him. And the reason why BITS was derived in the first place is to develop a standard that's supported by best practices. That means it basically becomes a standard that the community is building based on experience, rather than creating something from scratch. And as the comic here shows, normal standards, we have already, let's say, 14 competing standards. Oh, this is ridiculous. We need to develop a new one. And then in the end, there are 15 competing standards. So here, BITS was really a new way of doing things in trying to get the community together and to, with the community, agree on a way, okay, can we combine certain things already? Can we agree on one, for example, imaging forward as basis, but then build on this and make it accessible to everyone? And the evolution of BITS has been quite rapid. So there was a kickoff meeting at Stanford in spring 2015. And afterwards, a meeting at OHBM, the Organization of Human Brain Mapping yearly meeting in June. And then it was already introduced to the neuroinformatics community at the INCN Congress later in the summer. And already in September, they had a first release candidate and public call for comments on a written description of the standard. And a version 1.0 was published along with the introductory paper not that much later. So the first, I would say, start of BIDS was very focused on MRI. So it covered structural MRI and functional MRI, but by now it covers all sorts of imaging modalities, MRI, PET, EEG, MEG, and there are also extensions going on for covering animal data besides human data, covering specifically imaging genomics, connectivity, etc. There's lots of activity in the BID space. The credit here should definitely go to the people starting everything, and this was started at the Poldrack Lab at Stanford and was largely driven by Chris Gorgolewski, who was also the first author on the BIDS data paper that is shown here and was published in Scientific Data here in the end of, or it was received in the end of December and then published in the middle of 2016. And by now, if you go to the BIDS website, we have actually a few extensions where there also have been papers published describing the extensions. Here, the EEG BIDS on top, that was actually led by Cyril, and then also an IHE extension, an MEG extension. And now for PET, we have done it kind of in two steps. We had first worked on establishing consensus guidelines, and here's our consensus guideline paper published in 2020. And that is following up now by a paper detailing the standard for sharing PET data, and I'll get into that in a second. And also here for the genetics BIDS extension. So you can find all those references and publications on the BIDS website within the actual specification. And the goal of BIDS is actually to make the data more accessible to researchers. Now, why is that? Well, we have a problem. We're just, as you can see here from the COBIDAS MEG publication, we stole a figure. When we look at the number of EEG and MEG publications, for example, it is just going through the roof. We have so many techniques that we're using now in terms of imaging. And even though there are similarities in our experimental design, what ends up happening is that every researcher organizes and describes their data in their own way. So if you just hand it over to someone else, it's very hard to reproduce the experiments. So BIDS is a tool to make your data more accessible. Making it more accessible for yourself in 6 months' time. We've all been there that we need to reanalyze some data or derive an additional thing for revision. And you look back at the data folder 6 months later, and it's like, okay, let me remember what I actually did. And it's even worse when you have other people in the lab working with your data, new students, collaborators, and so on. It takes a lot of overhead to introduce other people to the data and how you have structured it. And finally, also, when we want to go beyond this and actually share the data freely with other researchers, then there are also issues. If I have well documented my data approach and my data organization in the first place, it's much easier to make this data accessible to other people. Now, I'll give you a very short introduction about the BIDS data structure basics. So the essence of BIDS is literally a directory hierarchy and file naming system. So it's as simple as this. So for example, on the left, we have our regular DICOM DR organization. You have this DICOM directory, you have some arbitrary series name as a folder above it, and then you have your single DICOMs. Well, if you actually know the series name, you might have an XLS sheet lying around somewhere who this series name actually was and what metadata is going along with the DICOM data shared here. Now, when we convert this to BIDS data, all we do is essentially resorting this. So already from the folder structure, I can see what's going on. So on the right side, you can see I could convert this to having a my data set, where on the top level already know something about how many participants I have in my data set. Then I here have a subject 1. I can directly see I have an anatomical, functional, and also in this case, diffusion weighted imaging available. And so in this case, it is very, very easy to see what kind of data I'm dealing with, how many individual subjects it is. And when we now look at a real bits folder on disk for about any or all modality, we can see that this is an example folder with the subjects in it. We could also have a source folder. It's not shown here, but where we actually put the original source data. BITS has decided to mostly base themselves on NIfTY. Almost all the imaging is saved in NIfTY files. And that means if you want to keep the original DICOM, you could keep them in a source folder that's separate with this data organization. There could also be a stimuli folder that shows when you're doing fMRI experiments or other experiments where you present the people with a stimuli. You could have that here along. Then if you have processed your data, there could be a folder with derivatives, meaning if I, for example, for PET, perform some kind of pre-processing, motion correction, partial volume correction, and then do quantification like kinetic modeling, then I could put those outputs in the derivatives folder. This way I have my raw data and my process data side by side. And the nice thing is this structure has been vetted by the community so much that it's so logical that it's quite easy to find your way around, because you have this rigid folder structure and naming convention. And this makes it also very, very easy for any computational tools built on top of this structure. Now, another very nice thing is that in every BITS data set, we have metadata files. They're saved as different text files, structured text files in JSON or a TSV format, where JSON is a more free text and TSV a tabular format. And they have standard dictionaries. That means, for example, in my data set description, I give the data set name, I say what version BITS have I used to sort it, I might provide a license, tell which authors are responsible for this data set, and so on. And this way, I can make even my data sets identifiable and citable. Furthermore, there's also a tabular file with the participants TSV. And here I can quite easily already at the root level see how many participants is this data set consisting of, what are their relevant meta variables like age and sex. So in this way, it's very easy to figure out basic demographics for a GA paper or when you're looking for specific subjects in a database. And the nice thing is both JSON and TSV, because of their structure, are human and machine readable. So it's not some binary you need to unpack that as a human is not accessible, but it is actually file formats you can just open on virtually any kind of operating system. And when we look also at the organization then within a single subject, for example, here our BITS folder on the left is subject 2. If we look at the anatomical folder here, we have a lot of name redundancy. And the reason for this is this way it's harder to make mistakes. And again, every imaging file, in this case a ZIP-NIfTY file, is coming with some metadata that describes the details of this NIfTY file. So inside the subfolder, we would have an anatomical folder, quite easy to understand. And if there would be an EG in a PET folder, it would be the same way. There would be, for example, a subject 002 something something EG in the EG folder or in the PET folder. So in that sense, it's very intuitive. There's a lot of redundancy, both for coding purposes, but also to ensure consistency when you're saving your data. And we always get a lot of questions about, yeah, but what can I then use BITS for? Or I don't quite see the usability in this. And as I've already mentioned before, it's not that you're imposing a new file format. It's not like you have to convert your data to this new imaging format that none of the tools you normally use can read. No, it's all based on accepted file formats like the NIfTY format for images and text formats that are readable by basically all major software tools, be it MATLAB, be it Python, be it even other imaging tools like ImageJ that you want to use. And it's also not in itself a database tool, it's a file organization. But once you have that organization in place, it's much easier to use real databasing tools like XNet or LORIS because they are already made BITS compliant. It's also not a search engine itself, but because you, for example, have metadata about the population that you have in your data set at the root level, it makes it very easy to make data discovery for search engines. For example, in the Google dataset search or in some of the data sharing platforms that I'm sharing with you in a second, for example, OpenNeuro or the Canadian platform. So in itself, BITS will not share the data for you, but it will help you and facilitate data sharing. And for yourself, when you look at it in 6 months, for other people in your lab, as well as for the rest of your community. And now moving on to PET. So this had existed for the MRI community, like I mentioned, since about 2015. And we came into the picture, I got involved around 2017. And when we're looking specifically at positron emission tomography, in short PET, it had actually a relatively good standard for a consensus nomenclature when we are talking about in vivo imaging of radioligands. And already back in 2007, this paper was published where people agreed at least on the modeling side of what to call things. Before that, everyone kind of named things differently, which also was an even, I would say, more fundamental problem. But with the standard nomenclature in place, we went ahead and said, okay, we need to drive this even further. We know from the publications, for example, with the Open Science Foundation, where people have tried to reproduce studies, that currently we are in a replication crisis. And in order to improve our replication records in science, we need to improve how we report on our experiments and also start sharing our primary research data. So in 2020, we went together with virtually everyone in the PET community, not quite, but almost, and came up with guidelines for the content and format of PET brain data, both on the publication side, so how do we report in manuscript what we have done, but also how do we share the data. This was work that was started by Guillermo Knudsen and Robert Innes all the way back in 2018 at NRM, and that then went on to get published later on. And based on this consensus paper, we then developed the corresponding PET-BIT standard. So we had already a consensus by the community of what information was the community asking for. And this was then merged about a year ago. And right now we just got the paper describing the standard in detail accepted at Scientific Data. It'll come out in the start of March. Until then, you can find it on the bioRxiv. And with now the standard, especially for PET being in place and for the other modalities following MRI, we of course want to share the data and share our primary research data. And currently, there is already a big platform in place for sharing neuroimaging data, and that is OpenNeuro. It's the official repository of the Brain Initiative and part of the Amazon Public Datasets project. It has hundreds of public datasets, more than 20,000 subjects. This is increasing all the time. There are about 10 to 20 new datasets uploaded every month, and more than 1,000 downloads are happening every month. So we have a lot of users going in and finding data on OpenNeuro. And we have spent the last year together with the OpenNeuro team to improve this and make it even more modality specific in order to make the search also easier for people coming from specific modalities such as PET or MEG or EEG. And in this way, we have established OpenNeuroPET, which is an extension of the OpenNeuro archive to really focus on PET data. And again, it follows the standard format and content that was developed both in the guidelines paper, but then also in the PET-BITS work. And on top of this, we are in the process of showing best practices when we're doing processing of PET data, as well as quality control checks. And the idea is with OpenNeuroPET that we want to really educate the community about the tools that are out there, about the possibilities with regards to data standardization, but also data sharing. And another goal is also that once people start sharing data, we would like to establish average images of receptor density for all kinds of PET tracers. We have in-lab worked on this a lot for the serotonin and GABA receptors, but when people share more data, for example, dopaminergic data, then we could really make for the community average images available that show you, for example, how on average would a normal person's dopamine system look like. And this could also much easier than be used by other communities such as the fMRI community. So in OpenPET, we will follow all the data standards we have established and also give some guidelines. And our benchmark for success are in the future the number of scans we will get, so PET data that will be deposited in the project, as well as of the resulting publications. Actually, it's become quite standard now, for example, with scientific data, that you can make data set publications, that you don't only have a scientific publication where you look at a certain aspect of your data and that is put out, but that you actually also publish a paper that describes a data set that you afterwards make publicly available. In that sense, it's much easier for people to reuse the data set that you're sharing. And we're really trying to engage the community in this, to provide help to convert data and also to upload it. And we're talking to all the commonly used software tools for PET analysis, for example, PMOD, in order to ensure that our data standard is accepted and can be used in all those software tools as input. The current setup of OpenPET is a little bit heavy on the Northern Hemisphere. We'd love to get collaborators from the Southern Hemisphere, but in Europe, it's the Neurobiology Research Unit and Rigshospitalet in Copenhagen. And then we have in Boston, the Martino Center for Biomedical Imaging involved at Harvard and Mass General, as well as the National Institutes of Mental Health in Bethesda, and then Stanford University, of course, as the basis of the original OpenNeuro. Furthermore, the whole initiative is split up in two parts. We have an American side where a PI is Robert Innes. He has a brain initiative grant at NIMH that is currently running, where we have Stanford Innes collaborator, as well as NRU. And on the other side, we have a European side that was funded through a private Danish foundation, the Novo Nordisk Foundation, that has been focusing very much more on European specific challenges with data sharing. And I'll get into those in a second. So when we're sharing data using bits, what we're aiming for is following the FAIR principles. We want to make our data interoperable. And this means that our data uses a formal format. And all of this is already given through our naming convention in bits, because this makes it very easy to be ingested by software tools. At the same time, because we are describing our data so clearly and also offer all the meta information necessary for it, we also ensure this way that our data is understandable. And finally, then, once we have the data in this interoperable and reusable format, we want to make it findable and accessible. And this is already now given for most of the data sets with the OpenNeuro platform. You can convert it to bits and then upload it. This way your data can be found and also downloaded again. The problem here is that OpenNeuro is not GDPR compatible in the current implementation. Brains are considered personal information, and thereby they fall under the GDPR rules here in Europe. But if you actually look in detail, and Cyril is the expert and will be able to answer more questions on this in a second, when we're looking at data sharing in the EU, we actually not only should follow the Declaration of Helsinki for good measure that tells us how to generate, collect, use, and process health data, but also the Declaration of Taipei that tells us we should also share our data. So for example, what we are working on to disseminate is that you could maybe consider using two separate consent forms, one for your study and one for data sharing of your study afterwards. Also, GDPR means you have some requirements to provide a lawful basis. So it means as researchers, we should actually have it as part of our aim to share the data. Often our research is funded by public institutions, and this means we should actually also provide an outcome in terms of the original data back to the public. And finally, within the GDPR, sharing and transferring is some type of data processing. So it's actually possible. There are mechanisms that you can, for example, contact OpenPET and ask us as a data processor to help you to facilitate data sharing for you. One of the proposed legal solutions that are already out there currently that you can find is the open brain consent. Cyril was instrumental in putting that together, where you can actually download example consent forms you can use in your study. And in this consent, you basically tell the people, okay, we're collecting your data for the purpose of their study, but they might also be used for other research projects in the field of medical and cognitive neuroscience. In this way, you strike a balance in the purpose of the reusability of your data, because you're not saying this can be reused for whatever, but it can be reused for relevant scientific projects. And this means also that you can still provide confidentiality of your data and, for example, cover cases like withdrawing consent, which are some of the rights that GDPR provides to participants. And with this, there's also a possibility under certain conditions to pass it on to countries outside the European Union. And furthermore, in general, it makes it easier to inform the people about their rights, because I think GDPR has brought a lot of confusion, both on the research side, but also on the actually the private citizen side. So here we have the community with open brain consent has worked on making this more accessible for everyone. And one of the things that is then still ongoing on the European side of OpenPET is a model that we could propose to make data sharing within an EU geographically constrained space possible. For example, if you have EU data, you can unfortunately not upload it to OpenNeuro, but what we can build is we can make databases within Europe that, for example, OpenPET could facilitate, that can then distribute, thanks to data-led metadata, to all the common web portals like eBrains, OpenNeuro, and CONP. So your data is findable worldwide. And as soon as users want to access the data, they then get, for identity and access management, referred back to a European site that takes care of everything and that actually facilitates the exchange, for example, of data sharing agreements and so on. So just to make all this sharing even outside of the EU easier. And we're working on this right now, where Cyril has been instrumental in writing a governance, and also in order to ensure findability and accessibility by sharing metadata with all the sites outside of Europe. And we're sticking to the BITS format with respect to the interoperability and the reusability. And of course, this way, we can also ensure real data protection for the participants, because we have access management and also data encryption. And of course, this can then also be audited by the authority, which is a requirement you now have with GDPR. That was our short introduction to all the efforts going on in the PET neuroimaging community towards open science and data sharing. OpenPET is one of the instrumental projects in this area. We have a lot of collaborators here. I try to put all of them up here. Cyril will now afterwards give you a short introduction to BITS, and we will be able to then answer any questions you have right after.
Okay, so that's going to be pretty short. I just wanted to follow up on Melanie's presentation on BITS and just to introduce to people here the ecosystem. So of course you've seen that BITS is about structuring your data, but it's actually much more. So first, to say that it's not just about data sharing, but it's also good research data management. Many of us are following PhD comics, and I like also this little cartoon, which basically, as everybody knows, when we started as PhD students or master students, we had a few things going on, and then quickly, you know, it increases, and now we're kind of way too many data. And so the nice things with having a standard is that it allows you to name and document your files, which the first thing is for yourself to find and understand the new and the old data just the same. Same for your PI as a PI or for students to get to work together without spending any extra time about explaining how the data are. Make sure analysis is quicker and you can share with others. And as Melanie was pointing out, I think the big reason why beads has been so successful in brain imaging is because it's a community standard. So there's not somebody else who come up with a way to do things. It's more everybody sitting together and agreeing, okay, let's do it this way. And it takes a lot of time. It takes way more time than coming up with something by yourself. But on the other hand, everybody agrees to use it. And so you can prepare your data going on the specification like this. And then another big thing with beads, which for anybody who would want to implement something similar to beads, which I think would be a very good thing for other areas than brain imaging, is to have the beads validator. So even though we not only developed the standard, but we also create a tool which allows you to say, yes, this follows the standard. Like in this example, this runs in your browser. Now, I wanted to also point out that this is not just biomedical, this is also biological data. I know that people here are also doing a lot of other things than brain imaging, or at least if it's brain imaging, not just whole brain imaging. So yes, we have MRI, EEG, and so on. But for instance, last week we merged the microscopy specification. And also soon we will be merging the animal electrophysiology extension. So which shows that there is many more than just whole brain human imaging. And actually, it's even more than whole brain because there's also an extension which works on other modalities and anatomical regions. And so that extension allows to extend to general medical imaging. And whatever extensions that you're interested to work on, I suggest you go on the website, and everyone and anyone is always welcome to contribute. As you can see, for this extension, when we propose extension, when people propose to do new things, everything is in Google Doc. So you don't need to be a coder. All you need to know is to bring your expertise in the imaging modality you have. And you can just read and you say, well, that will not work for me because I do this, I do that, and people agree on, okay, maybe we need to change things. Just to finish, I prepared a little slides that I put on the chat earlier. You can access on the website, which is just a quick recap on all the links that you can get about getting information about beads, preparing your data. For instance, we have converters. These are tools to take source data. So it takes your images and then it tries to convert it automatically into beads. And then you also have what I call beads grabbers and apps. So the grabbers are things that automatically read a beads dataset, and then that allows you to document, query, and so on. And of course, because everything is a rigid structure, you can create these apps. Those apps are going to completely automate the analysis of the beads dataset. I will just finish by again pointing out to the website. Thanks.